Hello everyone. Today we are going to discuss about the fundamental concepts of monohybrid cross. Before we discuss the monohybrid cross, we need to discuss the key terminologies associated with so we can understand it better. So first we need to understand what is a gene. A gene is a specific segment of DNA located on a chromosome that codes for functional product using usually a protein, thus influence a particular characteristic or trait of an organism. So it is a segment of DNA of chromosome, and we know DNA give rise to RNA, and it codes for protein, which gives to specific traits. So that's how gene controls specific traits or particular characteristic in an organism. Another terminology associated with is allele. An allele is an alternative DNA sequence of same gene. It is important to note that it is the alternative DNA sequence of the same gene located at the same locus of homologous chromosomes. We know homologous chromosomes, each gamete contribute to one chromosome, and one chromosome comes from the father and another chromosome comes from the mother. So an allele is an alternate form of the DNA of the same gene located on the same gene locus of homologous. Suppose these two are homologous chromosome, we can see, and this green one is denoted by one type of allele and red is another type of allele. So it is present on the same gene locus, as you can see, and it is the alternative form of the DNA. So they code for different traits. If allele present are different, then they are called heterozygous. If they are present same, allele presents are same, then they are known as homozygous. In diploids, each individual carries two alleles, forming genotypes capital A, small A, or small a in population. We count allele across all individuals constitute the gene pool. For each gene, suppose for each gene there are two alleles. As we can see, there are two alleles, one from each parent we receive in case of homologous chromosomes. Here is then we can see trait. What is a trait? Then here you can see it's a free earlobe and it's a attached earlobe. So lowest part of the ear is known as earlobe, which is attached to here. It is not attached to skin, and it is attached to skin. So it is a different types of trait. So these are ruled by the DNA, different forms of the DNA or alleles. So therefore each trait is there influenced by both paternal and maternal genomes, because both contribute to alleles which give rise to the genotype, and after this genotype is formed, this rise to the traits, which is also known as the phenotype. These traits are expressed in offspring depends upon the interaction between these two alleles. The foundational principle of this pattern of inheritance were first established through Mendel's classical experiments with Philipp Franz over a century ago. Genotype is the genetic constitution of an organism for a trait represented by the pair of alleles carried by capital A. Suppose it carries. Suppose there are three types of alleles. These are homozygous, capital A, or small a. If these both the alleles are same, then they are known as homozygous. If the alleles are different, then they are known as a heterozygous. The phenomenon is observable characteristic of trait and organisms such as height or color produced by interaction of genotype and environment. Genotype, when there is an interaction between genotype and environment, the phenotype or trait is produced. So now let's discuss about the scientist Gregor Mendel. Mendel was educated in a monastery and went to study science and mathematics at the University of Vienna. He went many Apart from his, many others have studied the interaction of traits in bees and other organisms earlier, but Mendel blended his knowledge of science and mathematics, was the first one to keep count of individuals exhibiting particular traits in each generation. This helped him to arrive at laws of inheritance. So he used the knowledge of both science and mathematics, and he kept the count of individuals exhibiting a particular trait in each generation. From those numbers, he derived the laws of inheritance. We'll discuss about it in detail. He has three laws: law of segregation, law of dominance, and law of independent assortment. In law of segregation, it states that each organism carries two alleles for a trait, as we discussed in case of diploid organisms, which separate during gamete formation, sperm and ovum formation, ensuring each carries one allele. So these alleles are separated during the gamete formation, and they again reunite during the zygote and development into the organism. And there is a law of dominance: when two different alleles are present, one dominant allele masks the expression of other. We'll discuss what is it in detail in the next slide. Mendel studied several contrasting traits in Piscean sativum, such as round versus wrinkled seeds, tall versus short plants, and white versus violet plants. In one of his key experiments, he crossed all tall plants with all short plants. Here we can see he crossed tall plants with the short plants. And in the F1 generation, the first generation consisted only tall plants. Here it is F1 generation, as you can see here. All tall offsprings are there with no intermediates or medium height individuals. This demonstrated that trait for tallness dominated over the shortness. So if there are two alleles, tallness is represented by capital T and shortness is represented by small t, then the offsprings will be capital T, small t. And here, as capital T is the dominant allele which represents tallness, tallness trait is dominated over the shortness. However, when F1 tall plants were allowed to self-pollinate, these F1 plants are allowed to self-pollinate, they are cross among themselves. The second generation, which is F2 generation, displayed ratio of 3 plants tall to 1 plant short. This result indicated that both tallness and shortness were inherited by F1 individual. Because if only tallness allele was there, then how come short plant is present here? So the allele which is represented for the shortness is also present. That's why it says heterozygous. Two different types of alleles are present, and when they are crossed among themselves, three tall plants are produced and one short plant is produced. From this, Mendel proposed that sexually reproducing organisms carry two copies of a factor, which is now termed as gene, for each trait. These copies can be identical. When they are identical, they are called homozygous. Alleles are identical, then they are called, when they are different, they are known as heterozygous, depending upon the parental contribution. The mechanism behind this is explained by the concept of gametes. When a gamete carries only one allele for each trait, because during gamete formation two copies of a gene segregate. When two gametes fuse during fertilization, the offspring restores the pair of arrays, one from each parent, as we have discussed earlier. Now, in genetics we use Punnett Square to visualize these numbers efficiently. To visualize this mathematically, scientists used a tool called Punnett Square, named after the scientist Punnett, British genetist who formalized this method. A Punnett square is a grid that arranges possible gametes. Suppose this is a female gamete. There are the possible female gametes are there, and possible male gametes are there. And when they fuse, here female is a homozygous and male is also homozygous. When they fuse, they produce heterozygous offsprings, as you can see here. For example, in a cross between two heterozygous tall plants, the gametes are capital T and small t from each parent. Arranging them in a Punnett square leads to the 1:2:1, and phenotypic ratio is 3:1 ratio. Phenotype is 3:1 because it gave rise to one homozygous dominant and two heterozygous dominant. So three tall plants and one homozygous recessive. So one short plant. This model not only explains Mendel observation but also establishes predictable pattern of inheritance. To confirm that F2 generation truly follows the predicted 1:2:1 genotypic ratio, Mendel logic can be explained using a test cross. Now we can discuss what is the test cross is. Suppose we draw a Punnett square here. So there are two gametes, capital T, small t, male gamete, and if there are female gametes, capital T, small t, then there will be four offsprings. First will be capital T, then there will be capital T, small t, then there will be a small t, small t. So we can see there is a one homozygous dominant, two heterozygous dominant, and one homozygous recessive. So they produce a 1:2:1 genotypic ratio. It is 1, these are 2, and this is 1. But phenotypic ratio will be 3:1 because these three are tall plants and this is a short plant. But how we know the tall plants are homozygous or heterozygous? How we know that? To determine that, Mendel's logic can be explained using the test cross. In test cross, what we do? The tall plants of F2 generation are crossed with homozygous recessive short plants. How it can help us to determine if the tall plants are homozygous dominant or heterozygous dominant? If tall plants are homozygous dominant, the offsprings will be tall, because if tall plants are homozygous dominant, if we cross it with homozygous recessive, the offspring will be tall. But if the tall plant is a heterozygous dominant, offspring will be segregated in 1:1 ratio, because if we test cross it with homozygous recessive, then we'll get, let's draw Punnett square here. This is a capital T, here is a small t, here is a small t, here is a small t. So here we can see we get two homozygous recessive, and which is small t, and two heterozygous dominant and two homozygous recessive. So the ratio will be 1:1 in of tall to short plants. But if we cross it with the homozygous dominant, then all plants will be taller. So to determine if the parent is homozygous or heterozygous, we test cross it. So test cross means we cross them with the homozygous recessive. If we cross them with homozygous recessive, if the plant is homozygous dominant, then all offsprings will be troll because it will be capital T small t. If we cross them with homozygous recessive and it is the heterozygous dominant, the ratio of tall to short plants will be 1:1 because two plants will be heterozygous dominant and two plants will be homozygous recessive. Suppose let us practice these two problems on monohybrid cross. In cats, black fur is dominant over white fur. Black fur is represented by capital B allele and white fur is represented by small b allele. Two black cats are crossed. Among them 8 kittens, 6 are black and 2 are white. Determine the genotype of both cats. Let us assume both parents were the heterozygous first. The both parents may be homozygous or more may be heterozygous. Suppose both parents are heterozygous dominant, then we'll get the genotypic ratio 1 is to 2 is to 1 and phenotypic ratio is 3 is to 1. Since kittens appear both black, uh, since white kittens appear, both black parents must be heterozygous because if both parents are homozygous, then only black kitten appear. Because if both parents are homozygous and they are crossed, they will produce heterozygous dominant cats and heterozygous dominant will have a black fur. But here it is written 6 are black and 2 are white. So they, since white kittens appear, parents must be heterozygous. Explain why some black cats can still carry the white allele even if they carry if they appear black. Even black cats can carry recessive allele B, which may appear in offspring if two heterozygous parents are crossed. So, if the black cats carry white allele, then they are heterozygous, and if they are crossed among themselves like this is a heterozygous parent and this is a heterozygous parent, then their offspring, one offspring will be uh, white and the three offsprings will be black. It will follow 3:1 phenotypic ratio. Let's practice another question. A plant with yellow seeds crossed with a homozygous recessive plant in a test cross. Among the offsprings, both yellow and green seeds appear. Determine the genotype of the yellow seed plant. So we test cross with a suppose we test cross with a heterozygous plant. The genotypic ratio will be uh, 2 capital Y small y is to 2 is to small y. The phenotypic ratio will be 1 is to 1. And if we test cross the homozygous parent, if the parent is homozygous, which is capital Y, all offsprings will be yellow. But here it is mentioned that both yellow and green seeds appear. Means the genotype of the yellow seed or plant was heterozygous. A second yellow seeded plant produced all yellow seeds when test crossed with capital Y, small y. As it is produced all yellow seeds when test crossed with the small y, it must be homozygous dominant. Because if we cross capital Y, small y, capital Y, small y offsprings will be produced and it will be all yellow colored. So from this we can know that the parent we crossed will be homozygous dominant. Thank you.